krama lite på din boll där. Ligger du bra som med armen? Funkar det? Ska den upp lite?
Giving blood can save lives.
Hejsan, hejsan.
Hej, välkommen.
Några blodgivare på ingång idag?
Det har vi.
Okej, kul. Bra.
But it has to be the right blood going to the right person. And this isn't always easily done because many of the properties of blood are still unknown.
Well, I see my role as kind of a matchmaker to find the best blood group compatibility between the donor and the patient. And we've taken the first steps on that road, but we still have quite a long way to go.
The only thing concerning Martin Olsson is understanding our unknown blood types. Via the red blood cells, we divide our blood into groups. We all know the most common types, but there are still a large number of blood types yet to be discovered. And as long as we don't know what they are, we can't always get a perfect match between donor and patient. This means that even today there is a risk for unexpected reactions with blood transfusions. The goal for Martin and his colleagues is to identify more blood types than the ones we currently know. Only then can the match between donor and recipient be perfect. And Martin has been working with blood for quite a long time now.
It was more than 30 years ago I started working here at the blood center, and there was something that gripped me immediately with the whole thing, and I haven't left the field of transfusion medicine since, working to try and improve the situation when it comes to blood group matching between donor and patients. What is striking is that everything remains pretty much the same since all those years ago. I mean, it starts with a gift from the donor, but even if everything is much more regulated today, this is the starting point for everything. What is equally striking to me, though, is that the whole downstream processing is so different. With the advent of genetic blood group typing and understanding of the blood groups, we can now match the donor to the patient much better. And that's where my research has made a difference, and we keep on trying to do that.
For many, blood transfusions are what keep them alive. Because when the body does not produce the blood it needs or loses too much of it, a patient becomes completely dependent on transfusions. One example of this is the case of Charlotte Stude from Malmö, who needs to get blood every third week, year in and year out.
Det är ungefär likadant varje gång. I början så, jag tror att de första gångerna som jag fick blod så behövde de inte kolla av lika precis som de behöver nu. Så nu får jag vänta längre innan jag kan få mitt blod.
Det är ju tyvärr ofta det som händer, att vi ser att man reagerar på de blodgrupper som finns och som man själv inte har. Och där anpassningen kunde bli ännu bättre. Och det är förmodligen det som gör att du får vänta, att vi försöker hitta ett blod som passar dig ännu bättre.
And this is also what parts of Martin's research are about, to be able to dive behind ABO and the RH groups in order to detect the genes with the help of DNA technology at the foundation of our blood groups. And with DNA typing, they can now establish a variety of blood groups, which has led them to finding more blood donors with these unusual blood types.
So blood is red because of the hemoglobin inside the red blood cell. Whilst most scientists so far, I think, have spent their time on investigating hemoglobin and its different variants, in our case, we've been interested mostly in the surface of the red blood cell and all the molecules that are present there, both proteins and carbohydrates. And we've also tried to find out what in our genes actually cause the different blood groups that are present in a population. And of course, after that, it's also very easy to wonder, why do we have them and what are the consequences of having one blood group or the other?
On the surface of our blood cells, there are a number of proteins and carbohydrates that have different functions. And small hereditary differences in these molecules determine which blood type you have. We have the well-known A, B, O, and Rh system, where you can be blood type A, B, AB, or O, and Rh positive and negative. But in addition to these, there are hundreds of other blood groups. One of them is MAM. But for Martin to research MAM is not that simple. This is the world's rarest blood type. Only 10 families on the planet are known to be classified with MAM negative, and only one of these people is a blood donor. But fortunately, with this particular blood type, Martin has managed to discover gold, a completely unique trait, meaning that one day we may be able to realize the dream of growing blood in laboratories.
For instance, when we took stem and progenitor cells from the rare MAM-negative patients, then it turned out that in cultures towards red blood cells, the formation of cells was much bigger. So we were able to harvest up to 5 or maybe even sometimes 10 times the number of cells.
Today, far too little blood can be produced and at far too high a cost. But by controlling the gene that controls growth, Martin and his team have taken a step forward towards being able to grow blood in the future.
So by analyzing blood and stem cells from these very rare MAM-negative patients, it appears like we've found the break on erythropoiesis, red blood cell formation. And of course, that is a great breakthrough for us.
In addition to the breakthrough with MAM, Martin and his colleagues have also discovered 4 new blood group systems, VEL, FORCE, SID, and GLOB, each of which have specific properties. And when it comes to blood research in general, this can lead almost anywhere and become important for all of us. It has been shown that our blood groups can affect different things, such as which infections we get, how easy it is to get a blood clot. They can even affect the risk of getting cancer.
What's exciting right now is to find out more about the newly discovered blood groups and see how they can work for us in developing new therapies for patients. But of course, we will also continue our quest to find all the new blood groups out there. We know they exist, and after all, that's what we do best.